forgive me. Stay here.
Swadika. Good morning, good afternoon, ladies and gentlemen. Good morning for those who are from Europe and Middle East. So my name is Rose Chitanwada. I'm going to be the moderator for this webinar today. So the webinar is on the gut health and feed. This is the webinar run by Informa Markets for the livestock and aquaculture vertical that we are approaching the industry by having the exhibition format, and we also doing the conference around ASEAN. You may see that most of our exhibition already moved to 2021 due to the pandemic situation, and we are worried about the safety of our exhibitors, visitors, and our partners that will have to travel to the event. And also with the readiness of the Livestock Taiwan. So we are going to have Livestock Taiwan in this November. So let's join in Taiwan if you could travel to Taiwan. So today we are going to focus on one of the hot topics for, I would say, for the last 20 years that I am in the industry on the mycotoxin. So the topic today will be about the implications of the low doses of mycotoxins for animal production and how to correctly manage them. So we have experts with us today here, Mr. Michele Muccio. He is the Regional Product Manager on mycotoxin risk management from Biomin. May I invite Mr. Michele to start the presentation, and hope you enjoy on the hot topic of mycotoxin. And if you have questions, you can put in the Q&A box. Hello, Dr. Michele.
Hello and good afternoon.
Yeah.
And a warm welcome to everyone from Singapore. I must apologize because the very moment I pressed the unmute button, Singapore was hit by a storm of biblical proportions, so you probably will hear a bit of background noise from the rain. However, hopefully you will hear me well. Let me share my screen. There you go. Hopefully you can all see my presentation. So as you can see, the title of this presentation is it's pretty much self-explanatory. So we're going to talk about low doses of mycotoxins and what we can do to actually manage them as correct as possible. For today's talk, I decided to split this PowerPoint in 3 parts. So we're going to focus on the low doses and what do they cause to animals. And we're going to have a second session on how to manage mycotoxins at the farm. So the strategies that you can use, that we can use on the field during harvest and also during storage. And we are going to have a brief parenthesis on mycotoxin sampling, as mycotoxin sampling is the foundation of mycotoxin risk management because it allows us to have a representative load of the dose that is in the field. And also we're going to cover the last point, some of the key facts about mycotoxin risk management, and I will also be quite brief on this part. Without further ado, I would say let's start with the first point. So we are talking about low doses, right? But what is a low dose? To be honest, this is not an easy question to answer because there is a lot of different information that is available on the literature. But generally, if we have a look at most of the publications, generally low dose refers to the levels which are below the European Community or the FDA guidelines, depending on where you are in the world. However, the problem is that those guidelines are actually not realistic of the real field challenges because they do not take into account synergism because, as I will explain a little bit later, mycotoxins undertake synergistic interactions. And they also do not take into account co-contamination, which is actually a consequence of synergism, or vice versa. So co-contamination is the conditions where a commodity, a crop, is contaminated by more of these metabolites, and the same metabolite can actually be produced by different fungi. So sometimes we end up with commodities which contains 20, 30, even 40 different metabolites. And this statement is not actually my statement, but it comes from the most recent literature reviews, which are starting to study more the synergistic effects and the co-contaminations, and they're actually reaching the conclusions that the guidelines that we have in the different countries are actually too high. So what should we do? Then, in my opinion, it makes more sense to speak about realistic doses. So realistic doses are what we can expect normally on the field, and maybe talk about low dose when we are below those realistic doses. So those are some of the common realistic doses that have been reviewed by the literature in the last year. This actually study from 2015 is a meta-analysis of different studies that have been done to mycotoxins, and those values here are expressed in ppm or parts per million or milligram per kilogram. So in Biomin, we usually talk about ppb, so part per billion, and to know the ppb, you just multiply those values by 1,000. And this also happens with the EU and the FDA limit, so multiplied by 1,000. So let me show you a bit how the mycotoxin situation looks worldwide before we dig into the topic on effects of animals. This is actually the most recent report that we have about Asia. So these are the samples that have been collected from January to June 2020. In Biomin, we analyze for the main 6 mycotoxins, so aflatoxin, Zearalenone, deoxynivalenol, T2, fumonisins, and ochratoxin. We report the percentage of contaminated samples, which you can see in this graph over here. On the right side, we also report the co-contamination because we have been studying the co-contamination for decades. So we have built quite a lot of knowledge on that. And we also report the average concentration for those samples in ppb, so parts per billion, and also the maximum concentration detected in a single sample. If we have a look at those data, you will immediately notice that mycotoxins that are produced by Fusarium fungi, so fumonisins, deoxynivalenol, and Zearalenone, so mostly mycotoxins which are produced on the field, are the most prevalent, with fumonisins being the most prevalent. And if you have a look at the contaminations, this is something that you can typically expect for 1 year. So we are talking about medium, in some cases, high contamination. However, let's not underestimate the importance of co-occurrence, co-contamination. So when a sample is contaminated by more than one mycotoxin. So in this first part of the year in Asia, we saw that 82% of the samples analyzed are actually contaminated by more than one mycotoxin. So it's quite a high number over there. I decided to include some of the slides from South America because I know that in some countries there is a tendency to purchase commodities from South America, especially from Argentina, Brazil, and the United States. So I thought that I would give you a sneak peek into those markets as well. So this is what happened in Argentina, and you can see a similar situation. However, in this case, we have a higher prevalence of Zearalenone and fumonisin, and also quite an interesting prevalence of aflatoxins, so quite high. And deoxynivalenol and mycotoxins are a bit on the lower end. Also, the co-contamination is around 60%, so more than half of the sample coming from the Argentina market, they contain more than one mycotoxin. Moving into Brazil, the situation remains a bit similar, meaning that we still see Fusarium mycotoxins prevailing. However, fumonisin become the most prevalent, followed by deoxynivalenol and Zearalenone, and we also see less aflatoxin in Brazilian sample. However, for fumonisins and Zearalenone, and fumonisin, deoxynivalenol, and also a little bit for Zearalenone, we can see some concentrations that starts to be a little bit on the higher side and could have some repercussion to production. Co-contamination is also quite similar to what we've seen in Argentina. So 61% of the samples contain more than one mycotoxin. So again, we are more than half of those. Last but not least, I want to share the situation in North America. And to be honest, it's actually a copy of what we already saw everywhere. So we know that Fusarium mycotoxins are the most prevalent. In this case, in North America, deoxynivalenol is the most prevalent, being detected in 81% of the samples, followed by fumonisin, and in the third place, we find Zearalenone. And also, if we have a look at the concentration, they're also quite interesting, especially fumonisins, but also DON and ZEN. Again, we have a co-contamination of 60%, so more than half of those samples contain more than one mycotoxin. Moving away from the Biomin mycotoxin survey, I want to show actually what are these mycotoxins doing to our animals, especially when we have them at low doses, so where they don't produce a very clear clinical effect. Well, the first system that mycotoxins encounter is the intestine, so the GIT, gastrointestinal tract. So this is the first barrier of defense by mycotoxins. And there are a few interesting things that happen in the intestine when mycotoxins are ingested. First of all, we know that the dose of mycotoxin that is being absorbed is mostly absorbed in the upper part, so 80 to 90%. And we also know that the intestine is able to absorb different mycotoxins at different percentages in different species. For instance, we know that in pigs, most of the mycotoxins are being absorbed. So we know that pigs are able to absorb more than 80% of aflatoxin, 65% around of aflatoxin, and also more than 55 up to 55% of the deoxynivalenol. Whereas for fumonisin, this is a lot lower. And poultry, we can see that it's a little bit more resistant in terms of absorption. So the absorption proportion is a little bit lower. However, these are, of course, results that have been reviewed by the literature. However, we have to keep in mind that we are talking about an in vivo system. So it's a very complex system. So it's also very difficult to say that those values are written in stones. We have a few factors that we need to keep in mind. One of them is the enterohepatic recirculation, which could cause the mycotoxins to be re-exposed into the intestine after being processed by the liver. And also, we need to keep in mind that this non-absorbed fraction, so the part of the mycotoxin that is not absorbed by the intestine will remain in close contact with the enterocytes. So it will act directly on the enterocytes of the intestinal cells, and it will cause all those disturbances like leaky gut, villi effusion, that we all know very well, because those are actually caused by mycotoxin directly interacting with the cellular layer. And I will go a little bit more in depth in the next slides at this point. And also, we need to keep in mind that mycotoxins are actually really, really small compounds, and they can really complex easily with nutrients and with other components that are in the gut that are together with the feed. And these interactions with components could actually facilitate their absorption. So their absorption could be actually increased in reality. So let's do not take those numbers in stones, black and white, but still they should give us an idea of what is happening. Most importantly, and this goes back to the non-absorbed fraction, to those mycotoxin being in touch with the intestinal cells, let's keep in mind that 70% of the immune response is in the gut, and the gut is the first barrier against the external stressors. This means that once we have compromised the gut, we really have a problem because we have destroyed 70% of the immune. Something that I mentioned before is that mycotoxins undertake synergistic and additive interaction. So again, I'm connecting back to the discussion that I made before with the co-contamination. And from the literature, we already know about some well-established synergistic interaction between some of the most known mycotoxins. For instance, in poultry, we know how deoxynivalenol and fumonisins can actually undertake synergistic interaction, so the toxicity of one mycotoxin can be increased by the presence of others. Also, deoxynivalenol and T2, they undertake synergistic interactions, aflatoxin and T2 toxin, and so on. And also in swine, we know deoxynivalenol with Zearalenone, deoxynivalenol with fumonisins. And actually, Zearalenone is very interesting because there's more and more evidence that Zearalenone can also undertake synergistic interactions with DON in poultry. So there is a few studies which are coming up that are actually shedding some more light on this. So as you can see, interactions within mycotoxin is very complicated, and it's very difficult. That's why it's very difficult to identify what is a low dose. And this is also why sometimes it's tricky, and everyone has a little bit their own idea of low doses. But I'm sure and I know that there will be more knowledge that will be shared in the next years. And I know that the scientific community is actually working more on studying cocktails of mycotoxins and their implications. So hopefully we will have a lot of a clearer picture in the future. Going a bit into the effects of mycotoxins or cocktails of mycotoxins on animals, well, we have to start from the gut because we started from the gut at the beginning, and this is the first system that mycotoxin encounters. So deoxynivalenol fumonisins, they are known both in poultry and in pigs to cause villi effusion and villi atrophy. These are just some interesting histological pictures. Of course, here we're talking about higher levels, but it's just to give you an idea what happens. So once we have a stressed gut, we have the villi effusion, we have villi atrophy, we have a reduced surface, which can be used for nutrients uptake and nutrient utilization. So we have less growth. And we can also know that the villi are actually responsible for captating the nutrients from the intestine and to transport them into the bloodstream. And we know that some of these mycotoxins, especially deoxynivalenol, they can actually disrupt the function of some of these proteins which are in charge of captating the nutrients, especially those proteins which are in charge of capturing the glucose. So we know that deoxynivalenol can actually silence them, and they can disrupt their function. So again, we go into the direction of the loss of nutrient utilization or the impairment of utilization. We also know that mycotoxins like deoxynivalenol and fumonisins, they can impair the tight junctions, and I'm sure you've heard this already many times. So the tight junctions are a network of protein, and they basically act like the cement that keeps the gut cell together, and they create this impenetrable barrier of the intestine, so they keep all the stressors, the pathogens, and the viruses out. But as you can see here, this is a peer-reviewed study. You can see these are the three proteins which are forming the network of tight junctions. Those are clodins, occludin, and ZO1. And you can see here you have on the bottom increasing concentration of deoxynivalenol and protein expression. So the higher the protein expression, the higher those proteins are produced. And you can see that when you increase the concentration of DON, and we are talking about low levels, the expression of these proteins fades away. So this means that those proteins are not produced anymore. This means that the gut is opening, and which is why we know that mycotoxins are actually predisposing factors for a lot of diseases, especially in poultry, necrotic enteritis, coccidiosis, and everything. So again, let's keep in mind that 70% of the immune response is in the gut. So it's important to protect the gut from mycotoxins, and there's plenty of evidence that mycotoxins are affecting the gut. Some studies just to show you how mycotoxins can be predisposing factors for necrotic enteritis. So we are here in poultry, especially broilers. So here, this is a study published by Antonissen in 2014. And you can see here, these are birds that have been challenged with Clostridium perfringens alone and with deoxynivalenol. And you can see here, you have a feed contaminated by Clostridium perfringens, and here you have the same, so with the Clostridium and the DON, and here you have the percentage of birds developing necrotic enteritis. You can see how this number is a lot higher when the Clostridium is together with the mycotoxin. So meaning that the mycotoxins, the deoxynivalenol in this case, and also fumonisin, because this study has also been repeated with fumonisin, you can see how they're actually working in synergy with the pathogen, making the necrotic enteritis more severe. The same has been observed for coccidiosis, so with exposing the birds to Eineria and cocktails of deoxynivalenol and fumonisin. This is a study, it's relatively recent, 2016. And in this study, they actually evaluated the lesions in the cecum and also the number of oocysts that were present in the mucosa and in the feces. And you have several groups over here. You have a control, which only contains the Eineria, and then you have the Eineria with the different mycotoxins. So you have DON, you have fumonisins, and then you have a combination of deoxynivalenol and fumonisins. And because we spoke about synergism before, you can see that actually the birds that received both mycotoxins together as well as the Eineria, they were the ones that developed the most lesions, and a similar trend can be observed with the oocysts. So with the shedding of the oocysts in the mucosa or in the feces, although actually the birds that were exposed to only fumonisin in this case had more shedding. So again, we are talking about predisposing factors, and as you have seen from the mycotoxin survey before, Clostridium mycotoxins, deoxynivalenol, and fumonisin are always present, and in some cases they can hit really, really high levels. So it's definitely there is definitely room for improvement in controlling them. This is just an overview of what we discussed so far of the necrotic enteritis story. So this is again is from the publication of Antonissen, and this is actually how basically deoxynivalenol it's causing like a broad array of problems. So here, as you can see, we have a healthy gut with healthy villi and healthy tight junctions. After deoxynivalenol arrives, we start having villi atrophy, villi fusion, so impaired nutrient utilization, and also the opening of the tight junctions. So when the tight junctions are opening, everything that is outside can enter, including pathogens, viruses, and other toxic entities, and also everything that is inside can go out. And usually those things that are inside are really valuable nutrients, which can be captated by the bacteria that are out there, and they can actually become an ideal substrate for making some of those virulent strains growing. So as you can see, you have a domino effect in poultry by having even low doses of those mycotoxins in the feed. Going a little bit more towards the immunity, I want to show you this slide. This comes from quite an old publication in 1984, but it shows you how mycotoxins are actually working in synergies with pathogens. So when you can have some of the effects which are triggered by the mycotoxin alone, but those effects, they can actually become a lot more worse when you also have pathogens. And what you can see commonly is increased risk of infections, more severe disease processes. Also, like some of the therapies become more difficult, the vaccination is impaired, and and so on. So generally, problems which are related to immunosuppression. And I want to elaborate a little bit more on this point by showing you some of the relevant studies that have been done in the last years, where it's actually shown that mycotoxins are actually helping pathogens to become more severe. This is a study where it shows the influence of DON on the colonization of the intestine by Salmonella typhimurium. And you can see, again, we are talking about very low concentration of DON in ppb. So we are going to 100 from 750 ppb. And if you remember my slides before, you see those numbers very, very commonly, even in the past years. And you can see that already going from 0 to only 250, you can see there is a rapid increase of the number of bacteria that were able to pass the intestine and to colonize the body. So this shows you how really mycotoxins can work as a gate opener for pathogens. This is also another paper from 2003 with the aflatoxin in broilers, which were challenged with the E. coli. And in this paper, they measured the mortality in percentage. And you have different groups. You have the E. coli only group, which reached in the study a 14% of mortality, but this jumped to 36% when you have both the pathogen and the mycotoxin together. So again, mycotoxins, they are really helping pathogen to become more severe. Let's remain a little bit with E. coli because I want to move to a more recent study. This has been published last year, and it actually shows you actively in poultry how deoxynivalenol promotes the translocation of E. coli. So you have different concentration of deoxynivalenol and a control, and the experiment was done for a total of 35 days. And you can see that at the beginning, already at day 21, you have quite an interesting colonization of the intestine in the jejunum and in the cecum, already from the group that received the smallest dosage of deoxynivalenol. So sometimes you don't need to have a massive concentration, and then you say, okay, no, I don't care. When the concentration is lower, I will not do anything. No, sometimes even a small amount of deoxynivalenol can actually be a big problem, and sometimes a small amount can make more damage than a bigger amount. And this is still the mystery of synergism, because we still don't know why it is happening. We also know that at day 28, we also have a higher colonization of liver and spleen in the group that received both E. coli and deoxynivalenol. So again, this is for also layer and breeder producer. This could be also something to think about. We also know that deoxynivalenol can be one of the many predisposing factors for PCO lameness as well, because it actually can favor the colonization of pathogens, of also the bones and so on. Let's move a little bit into pigs, but we still stay on immunity. I want to show you this study from 2006, where we see the effects of low dose of fumonisins and salmonella on pig heads. So we are still talking about interaction between mycotoxins and pathogens. In this study, we measure the lymphocyte proliferation. So in very simple words, the immune response. So the higher the immune response, the better the animal is responding to the challenge, the salmonella in this case. And we have 4 groups here. So we have F and S, where F is fumonisin and S is salmonella. When it's minus minus, it means that there is none of those. So the white is the control, no fumonisin, no salmonella. This light gray is the group that only received fumonisin but no salmonella. Then we have this white stripe is no fumonisin and salmonella. And then the gray stripe is both mycotoxin, fumonisin, and salmonella. And you can see, for instance, at day 7, so very, very quickly after the challenge, you can see that the group that only received salmonella, because it's this one, no fumonisin, actually was able to build up quite a good immune response against the pathogen. But if you have both the salmonella and the fumonisin, you can see how the immune response is actually very low, because those mycotoxins are immunosuppressant. They can act directly on the immune system, disrupting the function. So the animal, as a consequence, is not able to cope with the pathogens anymore. And that's why we go back to the slide that I showed you before about the synergistic effects between mycotoxins and the pathogen when I tell you that therapies and vaccination become more difficult. They become more difficult because the animals become immunosuppressed. And again, we are talking about doses of fumonisins which commonly occur on the field. Going a little bit into viruses, there's no best time of the year to talk about viruses than this one. This is a study from 2017, and is actually it discusses the effect of ochratoxin on the promotion of viral replication. And specifically, we're talking about porcine circovirus type 2. I just want to show you these two slides just to give you an idea. So again, we are talking about low, very, very low amount of ochratoxin. And we are talking about the production of this protein over here, which is a protein involved in the formation of the viral capsid, so one of the viral parts. So the higher this protein is formed, the more it means the better the virus is being efficient at replicating. And you can see that when you have the control, so no mycotoxins, you can see that actually the immune system is able to cope with the virus, with the viral infection, and it's actually being able to keep the viral replication low. But even by adding very small amount of ochratoxin, you can see how the production of this protein is actually jumps sky high. So meaning a clear sign of immunosuppression, clear sign that the animal is not being capable to cope with infection, and the virus is free to produce his own parts and to keep replicating and to keep infecting the animals. We can also see here on this graph on the right side, this is just shows you the number of infected cells by the virus, and you can see that compared to the control, and you also have the virus and the mycotoxin, you can see that this is statistically significantly higher when you have even very small amount of mycotoxin, in this case, ochratoxin in the feed. Because we are still talking about immunity, also vaccination. So I just want to show you how we are still talking about pigs, and we are talking about pseudorabies vaccine. You can see also here you have the antibody titer. You have a negative control with no mycotoxins, and you have the group that receive a combination between deoxynivalenol and zearalenone. Because I showed you before in the synergistic wheel that in pigs, even deoxynivalenol and zearalenone can actually undertake synergistic interaction, and they can cause this problem because all mycotoxins are actually immunosuppressive. So you can see here a good decrease of the antibody titers, and you can see this is a trial that we did with our products at the University of Berlin. You can see that actually because we have biotransformation in our product, we were actually able to recover those antibody titers by deactivating those mycotoxins. Last mycotoxin that I want to talk about is zearalenone. So zearalenone is often overlooked, but it's actually it's quite challenging for pigs, but we also have good evidence about poultry, and it produces a broad array of effects. So when we have a look at the female swine, so this is a table that summarizes the effect in swine. In female swine, we know about affecting the reproduction cycle, the conception, also ovulation and implantation. So it can be very problematic. It also produces embryotic death. So the survival of the embryos, it can actually be impacted by these mycotoxins. Some of the clinical signs, like the enlargement of the mammary glands and the swelling, reddening of the vulva, and also the rectal and vaginal prolapses. Those are things that we commonly see. And we also see them in piglets, because zearalenone can actually be passed to the piglets from the mom. So it can actually cross the placental barrier and also produce these problems in piglets. We can also see, if we have a look in other pathologies, could be the atrophy of ovaries and the hypertrophy of the uterus. So again, we are talking mostly about reproductive problems. So zearalenone is an estrogenic compound. It causes mostly fertility problems, not only in females, but also in swine, in male swine, sorry. So we can see, for instance, testicular atrophy, which is something that we also see in poultry. Impairment of semen quality in swine, but also we see that in poultry. And also we can see some direct effect on the reproductive organs. And I mentioned already about the piglets, what happened. This is again, is just to show you on the reproductive tract when we have Zearalenone, and we can show here the effects on the vulvar volume and on the weight of the reproductive tract. So we have a control group with no Zearalenone. Here you have the vulvar volume and the weight of the reproductive tract. And see when you have Zearalenone in there, how those two parameters are actually heavily impacted. So the weight and the vulvar volume, they're actually increasing. So you can clearly see the effect of these mycotoxins. Again, this is an experiment that we've done at the university also with our product. And because we can biotransform Zearalenone, we will actually be able to slowly recover this condition. Just to show you a bit of a picture how it looks like, this is from the same trial. And you can see here the control group, so the ones that didn't receive any Zearalenone. And you can see already here, if I remember correctly, we're talking about 500 ppb of Zearalenone, so concentrations that normally happen. But you can see already after a long-term, medium long-term exposure, how this one really changes. So you can clearly see when you're doing the necropsy evaluation, you can clearly see in some cases the effects of this mycotoxin. Because I also mentioned poultry, again, and we are talking about Zearalenone, I brought you here this study, again, from 2016. So this is a study which uses quite interesting levels of mycotoxins because it's a combination of aflatoxin and Zearalenone, and 260 ppb of Zearalenone are actually quite common, and so are levels of aflatoxin which are above 100 ppb, especially when we talk about more warmer and wetter climates like South Asia, Southeast Asia. This is just the effects of the synergism of these two mycotoxins in layers, and we're talking layers, so we're talking egg production and in general. So when we have a look at the whole egg production in percentage, you can see that compared to the control, here you have control, aflatoxin, Zearalenone, and the two mycotoxins together, you can have up to more than a 50% reduction in egg production compared to the control when you have the two mycotoxins together. Also, feed intake, when you have the two mycotoxins together, you can see how they were able to work in synergism in this trial. And you can see how the feed intake is a lot lower compared to the control and a lot lower compared to the two mycotoxins alone. It stopped raining, so hopefully you will hear me better now. Some other parameters that have been measured in this peer-reviewed studies were the egg weight, the eggshell thickness, and the eggshell strength. So going into the egg weight, you can see that it's been also decreased in the group which received both mycotoxins, and slightly more than the two mycotoxins alone. The eggshell thickness was not greatly affected by the synergism as for the aflatoxin alone, but the eggshell strength actually was. So it was more impacted in the group that received both mycotoxins compared to the control or the two mycotoxins alone. So again, you see also how it's difficult to evaluate synergism because it doesn't work the same with every parameters, okay? Some parameters, even in the same trials, in the same species, with the same mycotoxins at the same concentration, were still affected more than others. So that's why we are again to the discussion of developing suitable methods to study those interaction of these cocktails of mycotoxins and to come up with new guidelines which can be actually applied for the future, especially now that we are going into global warming, global training, and so on, and those problems like mycotoxins and so on, they will even become more consistent. Jumping into the second part, so managing mycotoxins at the farm. So I want to discuss the importance of GAP, or good agricultural practices, and also the representative sampling. As I mentioned before, no representative sampling means no good analysis, means not a good assessment of what is happening on the field, and also wrong mycotoxin risk management program. There are some things that we can do to minimize mycotoxins. So on the field, doing storage, doing harvest, and doing storage. So On the field, we can actually try to prevent as much as possible crop damage, because damaged crop means that fungus has easier access to the nutrients and can grow more on the crops. We can use resistant breeds, but there's still a little bit of is still not a very established territory, the one of mycotoxin resistant crops. So we still know that there is a lot of drawbacks with that, and it's not something that would be implemented anytime soon. Using proper soil cultivation, so tillage, and using the proper sowing dates. So by sowing dates, by doing tillage, it means that we are moving the soil and we are removing the particles from the previous harvest, which can become the ground for the growth of Fusarium. So we basically make it more difficult for the fungus to grow. Also, using the right sowing dates, it means that we're actually planting the plant at the proper stage, so the strength, so this plant will actually perform at the maximum of its capacity, and it might be able to have a certain degree of resistance. Performing proper crop rotation, reducing the plant density will also minimize the growth of fungi and mycotoxins. Using adequate fertilization, so not too little, otherwise the plant is stressed and it's easier for the fungus to attack, and not too much, because what is not used by the plant can also be used by other organisms like the fungi. Also, of course, performing weed control goes in the same direction of a reduction of plant density. If we're going to harvest, there are different things that we can do. So following an appropriate harvest time, so that we are harvesting the plant at the optimal physiological state. Controlling the moisture. So this is also very important when we are drying the grains, and also when we are storing the dried grains. So make sure that the moisture is as low as possible. Use appropriate harvest equipment, so something that is not causing damage to the kernels, or also try to clean the equipment after it's been in contact with the soil. When we are processing the grains and we are preparing them for storage, it's important to remove the damaged portions, because the damaged portions are more accessible to fungi as a nutrient source, so the fungi can actually grow easier on them. Also, the drying rate and the temperature, controlling them helps to have better quality grains and harder for the fungi to grow on them. And also avoid mixing of grains as much as possible. This also helps, so not to mix different grains coming from different fields, and also minimizing the growth, helps to minimize the spreading of spores and the growth of fungi. Doing storage, I mentioned already, taking care of the moisture, so using chemical control, so using, for instance, antifungal agents. Antifungal agents will not do absolutely anything against mycotoxins, but they might help to prevent the growth of storage fungi. So you will have to deal with less storage problems. However, this is a bit of a gray area, because for instance, in all the fungicides that are used, that are authorized in the EU, they're actually not strong enough against all Fusarium strains or against all Aspergillus strains. So because otherwise they would be too toxic. So if you're putting something in there that is actually destroying all the fungi, so you have no fungal growth, you also need to check, okay, how toxic is this fungicide? Okay, I'm preventing fungi to grow, so I'm preventing mycotoxin formation, but then I'm adding something that is actually maybe even more toxic. So a little bit of a gray area that deserves a little bit of research as well. Also, immediate drying of grain, of the wet grain, and try to store them without delay. So try to really dry them more efficiently as possible, and to store them right away. And also, the maintenance of appropriate storage conditions. So make sure that there's no water infiltration, temperature control, humidity control, also insect control. So make sure there is not a lot of access spot for pests to also go in and contaminate the grain. And also, of course, avoiding long-term storage. This also helps to minimize the growth of those mycotoxins that are produced during storage, like aflatoxin and ochratoxin. So again, we talk about management on the farm, and we talk a little bit about sampling. Sampling is very important, as I mentioned already before, and it is important to have a representative sampling. The reason is very simple, that mycotoxins, they are not evenly distributed on the lot, but they actually tend to accumulate in hotspots. So we want to make sure when we are sampling that we are sampling from every layer in the lot, and we are sampling from also every height. We are also sampling close to the walls of the silo, because there is where we have the water infiltrations and everything. After we have sampled thoroughly and we have sampled from every line and also from every height, we want to mix those samples that we have taken into what is called an aggregate samples. And afterwards, we take scoops from these aggregate samples and we decide how much we want to send to the laboratories, and the rest we can put it back. If we do like this, we will mix properly the grain, and we will have a 100% representative image of the contamination load in the feed. If we don't do that, we maybe sample from here. So we sample only mycotoxins and we're like, oh God, now I need to get rid of everything. Or we sample here and say, no, the feed is clean. I don't need any mycotoxin management. So, and you know, we know that this is not true. Then we know that we will have problems in the future. So by doing this procedure, we create a representative sampling. Representative sampling means a correct analysis of the mycotoxin load. If you have the correct analysis, then you can apply the proper mycotoxin risk management practice. These are just other examples on how to sample from silo, from truck, and so on, but they're pretty much self-explanatory. So we go back to what I said before: try to sample throughout the whole surfaces and throughout the whole heights. And also here I can show you something that comes from the European guidelines, AC 401/2006, on how to sample, and this is basically gives you the size according to the size of the lot. So in tons, so up to 1 ton, you want to take at least 10 samples, and you want to have an aggregate samples of at least 1 kilo. Okay, so it means that you take you take samples, you take at least 10 samples, and you try to have at least 1 kilo from 1 ton of samples. And of this 1 kilo, you can mix it again. You make you prepare your samples that you send to the lab, and then the rest you can put back. And then increases up to 10 tons, at least 40 samples, at least 4 kilos, and so on and so on. We are at the last part of the webinar. We have started a little bit late, so I think I still have a few minutes. So key facts about effective mycotoxin risk management. So when we talk about mycotoxin products, so first of all, what I showed you before about managing the mycotoxins on the field, doing harvest and doing storage, will actually help to reduce the contamination, but it will not eliminate the threat. Okay, if you want to fully get rid of mycotoxins, the most efficient way is to do it in the gastrointestinal tract of the animals using anti-mycotoxin products. The problem here is that the market is flooded by binders. So 98% of the products that you find on the markets are binders. Binders are substances that you add to the feed, and they are supposed to immobilize the mycotoxins and prevent them to being absorbed by the animals. Usually those binders are clay minerals, so you have bentonite, zeolites. In some cases you have yeast components, activated charcoal, and also modified clays, which are known as organ clays. Binders are the most common and the most cost-effective solution. And then you have a lot of mixed product also out there. So you have clay binders, which are mixed with vitamins, yeast, plant extract, and they claim multiple strategy for mycotoxin detoxification. However, there is like extremely little evidence out there that those products are working. When it goes to binders, they rely uniquely on passive absorption. This means that the binder needs to find the mycotoxins in the feed. So it also depends on the amount of binder, the amount of mycotoxins, and how is it mixed in the feed. And of course, they're not effective against all mycotoxins. If they bind, their adsorption is basically limited to aflatoxin. But even for aflatoxins, we know there is a lot of evidence that most of these products are also not able to bind aflatoxins. And there is absolutely no binding of other mycotoxins. This has been reviewed in the literature and has been established. When it comes to mycotoxin absorption, there is 4 characteristics that you need to keep in mind. So the specific surface of the binder, so how big is the surface? So in terms of like how much space this binder particle have to accommodate the mycotoxins, of course, the bigger the better. You want to make, has to be, the charge is the second parameter. So the charge between is what actually makes the binding stronger, because those mycotoxins are actually charged, the binder is charged, and the charges are tightening the bound, and these charges are very pH dependent. So when we go from low pH to high pH or vice versa, they can change and they can also cause more desorption. Also the CEC, or cation exchange capacity. This is actually the ability of the clay to store and release the cations. In simple words, is how capable is the clay to change its own charge based on the pH. So how also effective would be to retain the mycotoxin, and also the swelling. The swelling is the ability to accommodate water, so to increase the surface, and so to have a bigger specific surface. What is important, you see a lot of products there. This product has the highest specific surface, the highest cation exchange capacity, but the literature tells you, and not Biomin, but the literature tells you that none of these parameters taken individually is an indication of good binding. So the secret is to find a good combination of these factors, and there is only one way to know if a binder is working, is to test it. There is no other way. This is, I mentioned before, so it's been reviewed by several publications, and it's also in the EU regulation. Binders are actually an effective tool against aflatoxin, and the EU only allows legal claims for registration of binders for aflatoxin. We also know that it works for ergot alkaloids and to a certain extent to aflatoxin. But when we go to other mycotoxins, we know that binding is not an option. The reason is when you see aflatoxin, you have a look at the structures, and you see that it's actually a flat and polar structure. So it's charged and it's flat. So when it's flat, it means that it can fit in between the layers of a bentonite and the charges of the mycotoxins. They can, in simple words, interact with the charges of the bentonite, and the bound can be actually tight. In the case of the oxynivalenol, there is a little bit of polarity, but there is no planarity. So there is no way the oxynivalenol can actually fit in between the layers of the binder. And also this has been reviewed plenty in the literature. Here you have several binders which have been evaluated for the ability of binding aflatoxin B1 in vitro, so in the lab. Here you have the content of hash. So from high content of hash to lower content of hash. The higher the content of ash, the more clay is present, because when you burn it, clay becomes ash. And here you have the content of yeast, so from low to high. You can see that the moment you decrease the content of clay, the efficacy of these binders goes gradually down. And also when you increase the content of ash, you don't see yeast, you don't see any benefits in binding aflatoxin. So the products that have a lot of yeast and very little clay, they bind almost nothing. So the binding of aflatoxin is purely dependent on the clay. Also, the oxynivalenol, there is several literature review. This is a study from Professor Sven Dennike, and it also tells you that yeast products, so products that have a claim on the yeast detoxification of detoxification of mycotoxin through yeast, they actually do not work because yeast is not a binder. So it's not recommended to feed these to animals if you have a deoxynivalenol problem. You need to use the right approach. Zearalenone, this is the same study. Again, we are talking about in vitro. So we are talking about conditions which are favoring the binder because it's easier for the binder to work in vitro, in the contrary to in vivo, in the contrary to in the body, because we have a lot less disturbances from the nutrients, from the buffer capacity of the feed and so on. And you can see this is the same study as before for Zearalenone. And you can see that basically the binding of Zearalenone in vitro, so when the conditions are optimal, is maximum 40 to 50%. And in vivo, you can expect a lot less. So again, also Zearalenone, it's not a mycotoxin that can be bound, although we see a lot of claims out there with the binding of Zearalenone. There is plenty of evidence to prove that this doesn't work. So 20 to 40% in vitro is what you can expect. When you go into the EU, and the EU has a regulation on mycotoxin binding, so the EU or the EFSA, so European Food and Safety Authority, those are independent organizations which are actually imposing requirements on identity, safety, and the efficacy of mycotoxin deactivation products. So the deactivation of mycotoxins is actually regulated in the EU. So if you want to register something in the EU, you need to prove that it works. You need to prove that it works against a specific mycotoxin, and you need to be able to prove that it's also safe for the consumer, safe for the animal, and safe for the environment. Also, when you want to register something in the EU, you are not limited. You cannot only provide in vitro results, which could actually favor the binding, but you need to prove the efficacy in vivo. And you prove the efficacy in vivo by using a biomarker study to prove the mycotoxin detoxification directly. And it's also not possible to make a general claim. This deactivates all mycotoxins, but each component needs to be targeting one mycotoxin, and you need to prove that this one works. Why the biomarkers? As I mentioned before, in vitro data are not enough, and the biomarker can actually prove the efficacy in vivo by looking at the specific parameters, and also it allows you to rule out the the compensation effect. So, for instance, in some cases, people can improve animal performance as an effect of the mycotoxin deactivator. We say this one works because it improves the animal performance, but we know that this could be because of an indirect effect, so presence of vitamins, presence of antioxidants, immune stimulators, and so on. In the EU, this is not allowed, and that's why we use the biomarker, because the biomarker can prove that a certain ingredient is directly deactivating the mycotoxins. This means that if you buy a product that is being EU registered, you're paying money and you're getting something that is doing the job for what is it designed. So you need something to deactivate mycotoxins, you buy something that deactivates mycotoxins. There is no compensations and no mixed products and so on. If you have a look at the EU register of feed additives, you can get it, you can follow this link, or you can just Google it, and you scroll down until you see category 1M. In category 1M, is for all the mycotoxin products which are authorized in the EU. And as you can see, there is only the Biomin products in there. So we are very proud of this because we have invested millions and many years in research, and we have actually developed solutions against mycotoxins which are backed up by science and which are authorized by independent authorities. I reached the end of this presentation. I am a little bit late, but we started a little bit late, so I hope I'm fine. Just a few concluding remarks. So low doses of mycotoxins, I show you how they can actually harm animal production. So by directly affecting the intestinal health and the immunity, and there is already plenty of scientific evidence on that. We can reduce mycotoxins by having good SOP or good agricultural practices, but it's not possible to eliminate them. However, we can reduce them by working on the farm, by working on proper management. We also need to learn that sampling is the key to identify the mycotoxin load. And if we have the right mycotoxin load, we can also have the right mycotoxin risk management strategy. And if we have a look at the world of mycotoxin deactivators, we see binders are the most used products, but they are not a good solution to tackle multiple mycotoxins, as most of the mycotoxins can actually not be bound. I reached the end of this presentation. Thank you very much for your attention. And if you want to know more about mycotoxins, visit this website. It's a non-commercial website. It's scientific, and we have studies, we have references, and so on. I hope you enjoyed this presentation, and thank you so much for your time.
All right, thank you, Dr. Michele. And due to the time, it's very short now. I think we're gonna go through 2 questions only because we have to prepare for the next session that will be on the other topic. So the question I would bring to ask you, Doctor, it says, what do you think the reasons of high level of mycotoxin in feed or feed ingredients in Asian during January to June? Why this time high level of mycotoxin? You have any idea?
It's not a very simple question that we can respond like in 2 words, but it usually comes from we have seen that most of those mycotoxins are produced on the field, so a lot of these parameters are weather related, and also from where the different grains are produced. So maybe some countries they have particularly hot season or particularly rainy season with more damages in the plants and so on, so more growth of fungi. And maybe those caused a growth, an explosions of these field mycotoxins. And the storage mycotoxins, we also know that they attack prevalently those climates where there is that are hot and humid, and usually those countries which tend to do longer storage times. So to see those. So the environment and the storage conditions.
All right. So what I will do, I will give your email to the person who asked that question. Then you can give the recommendation to them. The next question, Doctor, is, do you have any reference on the safe level of mycotoxin in swine completed feed?
Yeah, I mean, there are different guidelines. So the FDA and the EFSA, they are published, and also like most of the countries, not most of the countries, but a lot of countries have their own guidelines for mycotoxins in animal feed. One very simple way to have a look is, if you remember my very last slide, I put this link to this mycotoxins.info. If you click there and you go on regulation, there is actually a summary of all the regulations worldwide. And then you can see what, because some countries are different, they say complete feed for pig, or some others is like a corn intended for pigs. So over there you can have a look, but there are guidelines around, yes.
All right. So Doctor, if I may ask, we will send many questions to you because the audience asks.
Yes, of course.
Reply directly to them. So thank you very much for your today presentation. Thank you to the audience.
Sawadee-krap.
Stay with us today. So the next session at 3 p.m. Thailand time, we are talking about black soldier fly larvae as animal feed in Asia. Let's hear the news about the replacement of the protein selection in feed market. Stay with us and see you at 3 p.m. in another 59 minutes. Bye. Thank you.